Welcome to part 2 of our revision video on DNA and protein synthesis. In this video, I'm going to explain the mechanisms behind protein synthesis. Remember that DNA is stored in the nucleus, packaged into large sections of DNA called chromosomes. Chromosomes can't leave the nucleus, but the ribosomes are where protein synthesis happens. So we need to make a copy of the gene that can be transported to the ribosome. First thing that happens is the DNA strand is replicated and a template strand is made. The template leaves the nucleus and heads to the ribosome. When at the ribosome, an amino acid carrier molecule can bind to the complementary template bases. Each 3 bases of DNA codes for an amino acid. These 3 bases are known as a codon. When the next carrier molecule joins, the amino acids can bond together. After numerous amino acids have joined together, a protein is made. Protein can then either fold to form an enzyme, or remain in long strands to form filamentous proteins such as collagen or keratin. We're now going to look at how mutations in genes cause a change to the protein structure. Due to this triplet code, 3 bases coding for a single amino acid, a single base mutation can change one of those amino acids in the sequence. In this example, a mutation in a single base has changed the second triplet from GGA to TGA. TGA codes for a different amino acid than GGA, and therefore the second amino acid changes. The change in this amino acid could affect the final shape of the protein. It may also be present in a non-coding part of the gene, cause no effect. However, if this mutation was in a coding part, if the protein was an enzyme, the shape of the active site may change, and the substrate might no longer fit. There can be more significant mutations occurring in a gene. If a base is inserted or deleted, it can cause a change in all triplets after the mutation. This insertion of a T has caused a change to the first triplet or the first codon. As a result of changing the first codon, all later codons have also changed. This results in a change to all amino acids after the mutation. This could have a significant effect on the structure of an enzyme or protein. Where we have a normal gene that has no mutation, it could form an enzyme that fits perfectly a substrate into its active site. However, a mutation in that gene for the enzyme can cause a change to the shape of the active site. If the enzyme shape is only changed slightly, the substrate may still fit, but it may not work as effectively. If the enzyme shape has changed more significantly, the substrate might not fit at all, and the enzyme cannot work. The active site may have changed to a completely different shape that fits a different substrate. This could affect the metabolism of the body as a different reaction is catalyzed. Here's our first exam question. Figure 1 shows one strand of DNA. The strand has a sequence of bases A, C, G, and T. How many amino acids does the strand of DNA in figure 1 code for? 2, 3, 4, or 6? Pause the video here and have a go. Okay, let's look at the answer to this question. What we need to remember here is that 3 bases of DNA codes for an amino acid. These 3 bases are known as a triplet or codon. Let's look at how many codons we have in this piece of DNA. We find here that we have 4 triplets of bases that would therefore code for 4 different amino acids. In this next question, we're told that syndrome H is an inherited condition. People with syndrome H do not produce the enzyme IDUA. Figure 3 shows part of the gene coding for the enzyme IDUA. We're then shown the strands from a person without syndrome H and a person with syndrome H. Strand K shows a mutation in the DNA which has caused syndrome H. The enzyme IDUA helps to break down a carbohydrate in the human body. The enzyme IDUA produced from strand K will not work. Explain how the mutation could cause the enzyme not to work. This is a 5-mark question. Pause the video here and have a go. Okay, let's go through the answer to this question. The first thing we should do is identify a mutation in strand K. You can see we have 4 triplets and 4 triplets in the other strand. The third triplet, there's a mutation of the base C to the base T. This has changed the codon from CAG to TAG. This mutation could have changed the amino acid that's present in the third place. A change in the amino acid could change the structure of the protein produced. As we are told that IDUA is an enzyme, the active site of the enzyme may have changed shape also. A change in the shape of the active site may result in the enzyme no longer fitting its substrate. In the case of IDUA, we're told that it's a carbohydrate. Let's take a look at the mark scheme. There's been a mutation, a change from C to D to T in the DNA code. This changes the amino acid. This could, as a result, change the structure of the protein. Changing the structure of the protein could change the shape of the active site. Change in the shape of the active site might mean that the enzyme no longer fits its substrate, or the carbohydrate. From this video, you should be able to recall a simple description of protein synthesis, explain simply how the structure of DNA affects the protein made, explain how a change in DNA structure may result in the change in the protein synthesized, and finally describe how a change in the protein structure may affect its function, such as an enzyme no longer fitting a substrate.